Benvenuti a GiovedìScienza. In un anno.
Welcome to GiovedìScienza, Science on a Thursday. And this is a rather special edition because 2015, as many of you already know, has been proclaimed by UNESCO the International Year of Light. Every now and again, UNESCO, which has as one of its goals that of promoting science and technology, organizes a year. It did so for the International Year of Astronomy, last year chemistry, and next year, 2015, is the Year of Light. But this has a lot to do with many different subjects. We will have one evening devoted to light, but this evening, to a degree, we will also be talking about light, because we'll be talking about fluorescence. Two of the three Nobel Prizes, or the scientific Nobel Prizes, this year were awarded to researchers who worked in light-related sectors. In the case of LEDs, it's quite clear. The Nobel Prize was awarded to the Japanese scientists, which is linked to light, but also the Nobel Prize on fluorescence microscopy beyond the ape limit has to do with this. In fact, we will be talking about it later on this evening. But I would like to mention two words on Giorgio Volpi. Giorgio Volpi is a researcher. He was born in Cuneo, but he works in Turin at the chemistry department of our university. And he is here because he won the 2014 GiovedìScienza Prize, and I think that is why you gave him a round of applause. Thank you very much, and thank you for the second round of applause. And at this point, it falls to me to explain what the prize is. This award was started 3 or 4 years ago, and it was with a view to promote good new science by young people. This has to do with a number of publications, their CV, their activities are all taken in consideration, but we also wanted them to be people who could popularize or communicate their results. And he is very much in this remit because he also has to do with teaching labs, so he teaches and communicates science, and in particular chemistry, and the technologies which in chemistry make it possible to prepare new compounds that have to be luminescent. We'll have a few experiments here in a few minutes. So I'm very pleased to give him the floor because he is an example of what Italian research knows how to do, and the fact that it recognizes and appreciates young researchers who are not very often recognized here. And he will be talking about the scientific activities which he's carrying out, something which is very easy in the United States and in the Anglo-Saxon world. Part of the funding of the research is devoted to communication of results, but in Italy this is not so, but we're trying to promote this trend. So thank you very much, Giorgio Volpi.
Va bene, allora passiamo al PowerPoint, così.
Here you see PowerPoint. We call it the versatile universe, and it has to do with the molecules that I synthesize, a universe of molecules that I prepare that are very versatile because they can be used for very different things. I come from the Turin Department of Chemistry, which is right close to here, and this is the sort of environment I work in. There are a lot of chemistry and lots of types of chemistry. I belong to the third type. I'm involved in synthesis rather than analysis and theoretical, and we create new molecules. We work with the other two, that is to say the analytical and theoretical one. For example, the Theoretical chemistry developed models to simulate and understand reality, while analytical chemists, for instance, quantify substances within a matrix, might be toxins in food, or, for example, determining the amount of mercury in tunny or tuna fish, while we're the ones who spend all day in a lab, and we prepare new compounds and study them. All I'll be telling you about today starts from this molecule. Its name is DPK, and it is available on the market. It has no interest in terms of luminescence or for any other applications, but on the basis of this molecule, first of all with my PhD, and now when I have time and I'm not working in the labs or for the teaching labs, I try to develop new molecules. This is a variation on the theme. It's a bit like music, variations on the same one. But before we start, I would like you to be aware of the dimension or world the chemist works on, because even in popularization, very often you confuse objects and dimensions. For example, cells and molecules are often confused. To understand that everybody understands and appreciates the dimensions that we work in, here you see. The human body is made up of this incredible number of cells, an enormous number, 10 to the power of 15. Each and every cell consists of molecules and atoms, and an estimation, because an eye cell is different from a liver one, but on average, they have a very large number of atoms, 10 to the power of 14, and a completely different scale of biologists, and as chemists, we manipulate very small groups of atoms. Here you see, in a glass of water, to leave the human body and use something that we all are familiar with, we have this incredibly high number of atoms, and our ability to, which goes beyond our ability to understand these numbers. If I eliminated 2 or 3 zeros, nothing would change, because we can appreciate between 1 and 100. And we have, you call, for example, 12 eggs a dozen, and a number like this is called a mole, or Avogadro's number. So this is what we need. These objects are so small that they're, first of all, invisible, can't be appreciated at microscopy, and unmanageable. And the most interesting thing is that our senses that have devolved to work on millimeters rather than meters cannot understand them or cannot see them. There are some events in the nature of chemistry, so I'm talking to the far left of this graph, that we have enormous difficulty in understanding. Here I've represented with images the things that we know. We all know what ants are. Each notch means that you're going to divide it by a factor of 10. So here you have an ant, 1 millimeter. So we've divided a meter 1,000 times, then again, and again. So it's a billionth of a meter, very small. How do you work in these dimensions? Because the problem of a chemist is to see, observe these dimensions. First of all, you were mentioning earlier on the Nobel Prize for Chemistry that has been awarded to the super resolution microscopy. We're talking about light microscopy now. This was the range, but with the 2014 Nobel, we've increased it by 2 orders of magnitude. But it's very far from what we as chemists manage, that is to say, small molecules. Right. So how can you see the object of chemist observation? It's impossible, but what we do is that we work with lots and lots of atoms, like you saw with a glass of water. Typically, what I do is that I extract, which is very similar to cooking. This is a heater. We put it within a balloon, which is connected with pipes to another balloon. Molecules, which is the ones I'm working on, work with different conditions and different reagents, and what we have at the end is a certain amount of matter that we hope is new, that is to say, a new molecule. We said that it's impossible to visualize each and every molecule to understand what there is within this mass, in this case a red one. We use instruments. The instruments are the means that we have as chemists to understand the inner nature of molecules. Each tool gives a signal, and each signal, this is something you learn, it can be referred to a small group of molecules. So if I see this signal, I know that there will be a certain group of atoms. If I see the others, I'll have another one, and so on and so forth. You will understand that all the analysis will be like a puzzle. Here we have, say, a bit of powder, and then we carry out tests in a lab with instruments. We put together, piece together the information, and if we manage, not always, we put all the atoms together, the ones that were suggested by the instruments, and we discover that in that powder there is an endless number or very large number of these molecules. And this is but an imagination or a description of what we imagined. This is what we've decided to use to describe molecules, but in fact it's very different from nature. Alternatively, we could do something completely different. For instance, in this case, we could take a molecule. The instrument gives us a signal, which is referred to a single atom. It's an isotope in this case, a rather particular atom compared to others, which our instrument is able to identify. And given certain conditions, the molecule will react, change form, change shape, and this will tell us that the signal, this is what I did during my PhD, we followed the movement of a single atom which changed shape and position within this that was a complex. And I did it for 3 times. So for 3 times it changed. Well, how long does it take? Well, we managed to do it by freezing the single positions. And here I'd like to show you how important cooperation is. I have my molecule. This is something that we did with an instrument. I had information. I was not sure of whether I had solved it correctly. I asked theoretical chemists that hadn't worked in the lab. They were sitting at their computers, but they created a model. These were the results that they obtained. These are models that they obtained with a computer simulating reality. Their results were consistent with mine, and this was a sort of test and a confirmation. When experimental and theoretical results are consistent, it means that probably we have reached an understanding. It's not always like that. If we go back, here we see I have developed a sort of tree of derivatives. There's different molecules, different variations on the theme. The first ones you see are the most interesting here. They are pyridinimidazoles. And last year we produced a series. Here you can see there's a sort of root, which is the root, which is the same in all molecules, while there is a segment up above which varies. The varying segment means that we have the different properties. So there's a common theme, a common root, and then there's a variation on the theme that varies. Which property? You can see it. It's luminescence and fluorescence. If we change a small part of the molecule, in fact, we change the ability of it to interact with light or how it interacts with light. So these are small changes, and then we have a range of variations, and some may be used for applications we'll see in a moment. To be sure that you have all understood and are aware of how the light and color of the objects are linked together, I'm just going to show you this slide where I show that white light is merely the sum of rays that bring the information of the color. Each ray is associated with a different color and a different energy, but beware, because if we consider the fact that light is but the ondulation of the electromagnetic field, and here we can have infinite number of wavelengths. We have very short wavelengths and long wavelengths. All that we consider light is that very small range, which is linked to what we said. We evolved as a species to survive in an environment where basically there are only those units of light that we call color, from violet to red. These have a wavelength that is expressed here between 400 and 700 nanometers. Interestingly and curiously, animals that evolved in different ecological or environmental niches can see other spectra. For example, many reptiles can see in ultraviolet that we can't, where we can't. So our molecules interact in this area because we can see that they're luminescent and colored. So if a molecule is colored, it absorbs light. And we as chemists are able to quantify how much light and what type of light it absorbs. We have graphs such as these, which are absorption spectra, and we say that for every wavelength that I send to the sample, how much does your sample absorb? And we get results such as this. Clearly, if the molecule absorbs light, we see it as if it were colored, because anything that isn't absorbed comes back to our eye, and the absorbed amount determines the color. This is true for all objects that we see as colored. The interesting thing of our molecules is that, well as absorbing them, they re-emit it. And this is why we can call them fluorescent or luminescent. When a molecule re-emits light, in the graph we have a second peak. And this is usually with a higher level of energy. This is the light that the molecule absorbs, and this is the one it emits. And when a substance is luminescent, it appears to be a bit like a light bulb. It absorbs in this area, but it emits light in the other area. We can show this directly here. This is one of the first experiments that I'd like to show you. Can we dim the lights, please? Here, I think you can see it like this. Here there are solutions that contain fluorescent molecules. In the normal light, they're virtually without a color. They're very diluted. This one might be slightly yellow and this one might be reddish, but when I switch this on, that has a light that gives a type of wavelength that they absorb, you see that they have colors. Then the sense that we're sending the wavelength that they need, and so they re-emit the light in this form that you see. Very simple. Maybe you managed to see them. You can see the first two are slightly colored, but they're not luminescent. But as soon as I give them the right type of energy, they come on a bit like lamp bulbs. This is luminescent and this is phosphorescent because they're organic molecules. And the light that this is ultraviolet light. Yes, this is ultraviolet. It's a Wood's lamp, and it has an enormous energy compared to colored light, the one we see, and the molecules have been organized so that they will absorb ultraviolet, and then we'll see the application and why we did it. Now, to explain in a slightly more theoretical manner what we've just seen, we can generalize and say that our molecules have a fundamental state. That is to say, when they're relaxed and happy, they're in this state. When the light hits the molecules, if they absorb it, and therefore they store energy, they move to a state that we call a state of excitement. So if they go from being relaxed to being excited, This, which I've represented, is the energy that the molecule has absorbed. And if the molecule has done this, then we say it's excited. Then the molecule will try and return to the fundamental state. It can do it in different ways. Very often it does it without light. For example, most of the objects we see are colored, but they are not really luminescent. Some may vibrate, may dissipate energy in one way or another, but when they release energy and get back to the fundamental state, they emit energy. You see the second arrow? And when they emit light, they go from the excited state to the fundamental one. Please notice what I said before, that is to say that the energy which is necessary to excite the molecule is much greater than the energy that they released. So they absorb a large amount of energy and release much less. This is why we lit the molecules with ultraviolet light, which is very energy rich, and then they released light in the range of the visible. So we dispersed the energy, if we can say that. Now, a chemist does something very different. We don't always work like chefs adding reagents. Sometimes we plan molecules. This is an example again from my PhD. We had a molecule with 3 portions and 3 parts. The first area, which is binder. This can bind to metallic atoms, while the second one was a molecular wire, and the third one, which we call a functionizable group, that is to say, it can bind to a substrate that could be a semiconductor or graphite. So to build, and we planned it, a molecule that contained 3 blocks with 3 completely different functions, where we had different chemical structures. To do this, there can be endless roads. So what we do is that we try and identify the best solution, which is the easiest and the most economical one, the cheapest one. Here we see I start always with my molecule. So we go to the end result, very low yield, 5%. And that's why I give them an amber light. We planned it, but a very low yield. A second attempt was a complete failure, while the third one, we turned it round, and we started the other way around, and we had the same molecule as before, but the yield was 43%, which is very good, and in this case, we got a green light. This manner of proceeding can last a month or 2, so we understand the level of frustration for us involved in synthesis, because you see examples where it didn't work. But the important thing in research is to understand that each and every mistake is something that teaches you. And I confess that very often there's also an element of luck. Even though Edison, that I quoted here, says that luck is only for stupid and incompetent people. But in fact, in science, this is quite useful when you know that Edison, to find a material for the filament of the lights, the one which are banned now in the European Union, but to find that filament, he tried everything. He tried something like 1,600 different materials and combinations. And he said once, it's not that I made a mistake 1,600 times. I found 1,600 solutions that didn't work. Well, me, I only needed 3 attempts, and not the 1,600. This is one of the ways in which you can develop. Now, when you find the right solution, the easiest route, what you do is that then you have something a little bit like stamp collection. You get a result, fine. At that point, you have different variations, changing something. And so you have, at this point, a range of molecules. And you can see it, even if you haven't got any chemical knowledge. You see that each molecules are very similar. I don't want to describe in detail chemical structures. But you can see that these formulae are very similar. They only change in terms of very small portions, and the ones which change are the ones that determine different properties and can then be applied. So once you found the right solution, a researcher can try to obtain as many products as possible. And these are some of the results. Going back to the initial result, here you see this was 2 weeks' work. At the last passage, a bit when you have a flambe in the kitchen, the molecule went flat, decomposed. This was unstable, and instead of having 3 blocks, it broke. Very great frustration, but you continued. We started from our original molecule, and in the end, I got a molecule that had 3 blocks, and we used it. This is how you develop it, and very often how you have to have attempts and repeated attempts. Now, I hope I explained how research in a lab of an experimental chemist works. I'd like to show you the possible applications in terms of engineering. I make molecules. I get this powder, this colored powder, and then I want to use it for something. We, as chemical researchers, are not the ones who are going to apply it or develop them or develop the structures, but we have to be aware of what they can be used for. And this is what I want to show you. In general, we should know that luminescence and fluorescence is something that surrounds us in everyday life. Here you can see some applications: plasma televisions, lamps, biomedical applications. This was not so in the past. Fluorescence is something which is not very widely spread in nature, and human beings didn't know it. The first ones are linked to Boyle's research. He was a very famous scientist who spoke about servants who were afraid because they eating sugar, which is something which we call triboluminescence, and they saw light in their mouth. But at the time, it was considered a bit satanic, something that had to do with spirits. Well, we know that it is a classical luminescence if you break a crystal. But this is to say that it was absolutely unknown. In the 17th century, there was a cobbler living near Bologna. He was using some metals which contained barium, and by chance obtained a fluorescent material. He had no knowledge, but it was a barium stone, which was called the Bologna stone, which is fluorescent at night. Those of us who use, for example, plasma screens, mobile phones, LEDs instead of the old lamps, are aware of it. The molecules which we do, and the reason why we built them, is this. This is the application which is called Oled, OLED, and it's a technology which is for light emission. We have understood that biomolecules interact with light, and as I showed you before, there were molecules that absorb light and release it, while these are within an electric circuit. And when there's electrical energy, what happens is that the molecule acquires energy from electricity, steals it, and releases it as light. So here you can see that there's energy, electric energy, and there's colored light. By modifying the structure of the molecule, the intensity and tone or nuance of a color changes. So this is a very low consumption compared to the screens that we have, because here behind the screens there is a lamp which emits white light, and here there are liquid crystals. But the light that's behind it is a very high energy consumption, while with this type of consumption there is a very low consumption of electricity because it directly excites the molecule so that it emits light. To give you an idea, a light bulb throws away about 95% of light, and so it transforms 5% into light. What's the efficiency here? These are prototypes. so it's not yet sure. We'll see what will be released in the next few years, but at least 10 times as much, yes. But this is not the only interesting aspect, but also that in the future it will be possible to create screens that can be flexible, transparent when they're not on, and when you have electricity going through it, the area where the molecule is comes a light. This, for example, is a prototype of a screen that is totally flexible and is completely transparent when it's off, and when it's on, here you can see the various tesserae where the molecules are, and it can form an image. A bit like a dynamo that's seen the other way around as an electric motor. That is to say, well, we all know what dynamos are. If I turn them around the other way, it works, but differently. The same thing happens with molecules. They can take light and transform it into electricity. Let me show you now some applications where molecules can be used to transform solar energy into electricity. The first one is an indirect method. We all know what the silicon panels are. They're devices that transform the sun's energy into electricity through the photoelectric effect. Light falls here. You can see you have a creation of electrons. These are injected in the electric circuit. This generates electricity that, for example, will switch a bulb on. And these are the devices. We saw it earlier on. Light from the sun, which is used by silica panels, is not uniform, but it is made up of different tones and different intensities, which we give a different wavelength to. And unfortunately, both the sun, which emits these rays in different ways, and silica panels. What you can see here, white, is the sun spectra. You see it's virtually continuum. There are areas that correspond to what our atmosphere absorbs, and the silica panels manage to convert all the colored part, while we lose the other part of the sun, which is the infrared, and unfortunately we lose also this area, which is the ultraviolet. The ultraviolet area, which arrives at low intensity because otherwise it would create enormous problems, but it is a region that has a very large inherent energy which the panel cannot convert. Furthermore, it damages panels because many components are degradable in ultraviolet light. So our idea was very simple. We put on the silica panel a small film. They seem transparent plastic sheets with our molecules inside. We saw it with Wood's light. It absorbs ultraviolet, and then bioluminescence, it emits color. Initially, it seemed to be negative because it was a degradation of energy, but here it's very positive because we absorb ultraviolet, which could not be converted into energy. We protect the photovoltaic panel because it causes problems to some of its components, and we transform the ultraviolet in useful energy. That is to say, we filter it and transform it into colored energy. Here you can see the violet energy going through the panel becomes colored light, which increases the yield. These are the devices that we created in the lab. This is a small film which I'm going to show you here. So you see there are no tricks. We use the same lamp, the ultraviolet one. Here you can see these are some of the films that we obtained. I'm going to position them here randomly. And you will see that they are absolutely transparent in the light which we have now, which is all visible light of that range. You see it? One is already lighting up because it's extremely functional. When I switch wood lamps on, they're not reflecting, but they're absorbing ultraviolet light, and they're transforming it into visible light. So the light which comes from the lamp here, which the silica panel could not transform, but the light which is emitted by these can be converted. So if the sun were here, we would have protected the silica panel below, and we would have increased the yield, the conversion yield. So using very simple things which had this property that was studied. Here, going back to the presentation, a last variation on the molecular theme. On the molecule I was using, in this case we have molecules that have this portion that is able to bind a metallic atom, and we bound these to iridium, which is a very rare and noble metal, and that enhances it. These are coordination complexes, because it's our molecule with a single metallic atom. We did it with iridium and rhenium, and every time that I attach or bond a metallic part, I go from fluorescent to phosphorescent, which changes the properties and increase the range of applications. In fact, instead of having silica panels, in this case, we convert it with a new type of technology, which is called Graetzel cell, and you can see it here in the image. These are colored cells where there is no silica. It's all a dye, and it seems to be basically very colored glass. These that seem colored glass are photovoltaic panels that filter light and transform it into electricity. This, which seems to be a glass panel, is an electric power station, a very beautiful one. In this case, it's no longer the silica, but it's a color or dye that does it. And it's completely separated from fluorescence, but the principle is exactly the same. Here it's represented by the red bar, and I'll tell you what is happening here. The color is this, and the green one is the fundamental states that we saw before, while the red ones are excited state. When the sun rays go and the molecule absorbs it, then the electrons skip or jump on to the excited level, and they're promoted to the other state. Without emitting light, they're then injected to the electrical circuit. Here we have a semiconductor electric circuit, and here we have the light that represents the energy that we have generated from the sun. So this is a closed and complete circuit, and to operate it requires the fundamental levels in a particular area and a well-defined area for the level of excitement. With a large range we found some molecules that are better than others. I would like to show them because since Professor Viscardi's collaborators have prepared some, I would like to show you them so you will understand which prototypes we're working on. They're very small, and you can see within them there are substances that are colored that are, in fact, dyes, green, red. Very often we also use myrtle because it contains dyes or colors, and we can transform it into luminescent energy. Are these stable molecules? Can they do this for a long time? Well, the development of this type of technology is to find molecules that are very good at transforming them, have a good yield, and be stable. Currently, we have results that we will comment with a graph, and there are other advantages which I would like to stress. But first of all, I would like to show you a cell here that we have connected. Here. This is the cell. I hope you can see it. It's red. And this is the potential that it's generating because it uses light, the light that comes from those lights above. And you see if I remove the cell, it goes down to 0. If I connect it, then it does things. There's no trick. Otherwise, you might think there's a trick in a battery. So a small device such as this develops an electric potential. If we put lots together, then you can have a large amount of energy. Going back to the presentation, I would like to show you what the current state of the art is as far as the conversion of solar energy is concerned. It's a very hot topic. Here we see over years, over time, the technological ability to transform solar energy into electrical energy. Each line, each colored line, has a different type of technology. Silica panels, which have a certain color. Then the ones that I've shown you, these are the ones, the orange ones. What do we understand from this? That they have a much lower yield than the silica panels, but they may have some advantages. I'm working on them. Well, they started much later, and the advantage is that they are very cheap, very, very cheap. So we have to understand that, yes, we have to transform as much light energy into electricity, but we must also have cheap devices, because if we want to put panels in a third world country, they can't be too expensive. Clearly, it is different if we want to have panels for the shuttle that have to transform everything. They must be very, very efficient. So the area of the graph here that you see up at the top represents a type of technology that creates panels that consume more energy in production than the one that they will produce in their entire life. So if I want to develop panels on a space station, that's fine. But if I want to use them on our planet, in our houses, I must do something much cheaper, easier to do, and easier to develop. Interestingly, also, to see where it begins, because it starts from 1975, but in fact, you could go back to the 1800s. All starts from the photoelectric effect, which was discovered by Becquerel when he was 19, and we started with 0.6% conversion. That effect was explained theoretically by Einstein 120 years after, and we'd completely forgotten the ability that scientists had of converting light into energy. This field of study was something that was only interested in those who were studying photography. For the entire 19th century, the interaction between light and matter, Daguerre studied, or others like that. They'd understood. They were amateurs, basically. They weren't even except they'd understood that light interacted with matter, and in the case of photographs, they have reactions, but when it absorbs light and energy, then it can release it in circuits where light energy was transformed into electricity. Here, you can see in this slide represents chronologically our latest product. These are the molecules you saw before, but duplicated. That is to say, we increased the symmetry. This year we created these products, which have the big advantage of being able to surround metal centers, creating a molecular plane that surrounds it, and the application for these is completely different from the ones you saw before. And clearly it has to do with light, because this is what my molecules do, and it's a biomedical one. That is to say, we wish to make, and there are some molecules that work that are very similar to the ones that we've developed. We've developed drugs or substances that interact with light and do what you see now in the video. The light goes there, it's absorbed by the molecule absorbs light energy, transfers it to the metal. You saw that radiation. The central metal detaches a portion, we lose a vector, a molecular portion. The key is in the one we lose, because it can be a toxin, a drug, it could be a bioactive molecule in NO or CO. So imagine that the initial molecule is stable, and we inject it in a patient. In this case, a sick rat. The molecule is stable. When I shed light on it, and the organic substances, such as the one, such as in my hand, are partly transparent, especially red. When you put a torch on your hand and you see that your hand becomes red, the violet and blue don't enter, but red enters. Now, if my molecule is able to absorb that light and do what we saw before, where I shed light on the mouse, here I am able to obtain what you saw nanoscopically in the animation. So I can release my vector. If my mouse underneath the epidermis had a tumor and I'm releasing a toxin, I will kill these cells only in that area, have selective killing of the organic tissue where there was focalized light. Let me release a drug, I do something else, and this type of treatment is already used topically, not within the body, because there are the problems in maintaining these molecules stable, but for some type of fungi that are very difficult to kill, we use drugs of this kind. You put them on the skin, and then you put a mask, you put light, and you release the toxin and kill everything. Future applications on the human body is this one here. You see it's in 3D. There's a human body rotating. The molecule that was injecting and that has been selectively lit up. Here you have a photoreaction, release of the drug, and clearly we know this. It's also a fluorescent molecule. So once the fluorescence is enacted, the toxin will then be used and expelled. With these 4 applications, it's an incredible range, and I would like to thank you very much for the attention. First of all, at least I got this message. In the work that you do, there's a lot of chemistry, but there's also physics, and there are also some which have to do, applications which have to do with biology, and then this technology, because when you, say, generate photovoltaic panels of this type, which, for example, could be used for advertising with this type of panel. Oh, yes. There are many things that are interacting. The boundaries between and among the various disciplines are blurring. And possibly the most interesting results are at the point of intersection. I'm thinking of this because I was talking to Professor Marchisio, and rightly he was saying, he is in the scientific committee that organizes GiovedìScienza Science on a Thursday. He was observing that this year, not only this year, there's not much biology, and this is something we'll have to redress. But it's interesting to notice that very often disciplines overlap, and so biology emerges when we talk about chemistry and vice versa. If I may, I'll just tell you an anecdote, because it's true, but it's also very difficult because worlds don't communicate much. If you work in the Department of Chemistry, you work with that. A few months ago, I went to talk about these things to the Turin biology department. They use fluorescence for microscopy. I didn't even know about that, but seeing their presentations, they do some very interesting work where they follow communication between plants and fungi, the symbiont organisms that use hormones to communicate. They attach probes and they communicate. So as a result, there will be a cooperation amongst us between the 2 departments, because they were explaining the problems of these fluorescent probes that we can solve quite easily because we're chemists, so we know how to work on this and how to work on molecules. And we, on the other hand, suggested that they would use phosphorescent probes. They'd never even heard talk of, they didn't know what they were, and they didn't know that there were different properties by simply modifying molecules. This was absolutely random. It happened while having a coffee before the symposium, and that's where you get good ideas with coffee. And this brings us, and I would like to brings us back to the GiovedìScienza, to the prize, because and I invite you to come to because there are 10 people who are shortlisted on the basis of publications and things like that. Usually 70, 80 people apply initially. So as I said, 10 are shortlisted. in an objective way, looking at their curriculum and looking at the publications. They're all people who have to be under 35, so they're really young for the Italian standard. In fact, they're very young for Italy. Maybe abroad they wouldn't be like that, but things slow down, things in Italy. Well, these 10 people who are shortlisted then discuss for talking for 6 minutes and 20 minutes with 20 slides. So 6 minutes and 20 seconds means it's more like 100 yards rather than a marathon. You have to concentrate everything in 6 minutes. At the end, there are 100 students of the high schools who come here and judge the contents, but also and above the ability to communicate this information. And so you get the winner. It's something interesting, and the person who wins has a small prize. But I think the real value of the prize is that 10 researchers, very often in 10 different fields, meet. They meet, they talk to each other, they each present their results, and usually the thing that one presents is a surprise for the other 9. So there are many surprises for many researchers. There's no coffee, but maybe we should have a coffee. And I think that that is something that we should be able to explore all together. If we as a CONVERTION, GiovedìScienza scientific committee, if we also, in memory of Aldo Fasolo, we will be talking about, to remember him, if we managed to do this, to have this interdisciplinary activity, I think we would do something useful and something which is nothing new, but which is not done a lot. In Italy, you do it, and I think maybe in Turin we could start doing it. I would say I have 1,000 questions, but we have a little bit of time, but not much, and I would like to put the lights on, hear what the public, the audience has to say. There are no questions, I have them, but let's see what your questions are, so that we can shed light on new details after this very interesting presentation.
Volevo sapere, una sostanza è fatta solo da un tipo di
What is a
molecola
substance
o da
made by
diverse?
one type of molecule or several? I am asking this question because I have 2 drugs, and when I asked which one had less cortisone, I was told there are different types of cortisone. They have different molecules, and I'm surprised. I thought that a substance was made out of one type of molecule. So thank you for the question. It's very difficult to understand if you're not a chemist. Pure substances have only one molecule. For example, if you have water in a glass of water, there are only and exclusively molecules of water. Well, we hope. Yes, if it's pure water, then there can be traces of mineral salts. But in the cases of drugs, I don't think you'll ever find a pure molecule. There are inactive ingredients, excipients, and several active ingredients too. As chemists, we have a dogma. That is to say, we work on pure substances, because if we mix different substances, then we don't understand the properties, which one belongs to which. So we work on pure substances, that is to say, with large amounts, which may be a spoonful or a jarful, with a single molecule, which is the same and which can be repeated. But in our everyday life, I assure you, it's very, very difficult to deal with pure substances, because nature tends to standardize everything. So in sand, which you have on the beach or the sea, there are very different types of minerals. In the Coca-Cola that we drink, there are acids, sugar, colors, and so on. Anything that you buy is probably a mixture of different substances, from sprays to paint, unless you buy a canister of CO2, in which case there's only CO2, but normally it's a mixture of different molecules.
Prego.
Volevo sapere esattamente che differenza c'è
I
fra
would like to know exactly what difference there is between fluorescence and phosphorescence, and what relationship there is between them and what relationship with radioactivity. And I would like to ask you what fireflies have. Well, it's a very subtle difference, and it took us quite a long time to highlight the difference. The two processes together, fluorescence and phosphorescence, are defined luminescent. When you don't know, you say luminescence because it has the two together. Now, up to about 20 years ago, the difference between these two was linked to time. That is to say, very fast phenomena were fluorescence, while longer-term ones were well, we were talking about before, if you put tonic water under light like this, as soon as you turn it off, there's no more luminescence, so it's fluorescence. If you take your dial and you can see it at night, that's phosphorescence. And now there is a difference. It's a time difference, but it's more subtle because it has to do to fundamental or excited state. We talk about fluorescence when the excited state is in single. That is, the electron stays up and stays unaltered. That's fluorescence. If the electron was like this and it jumps, and that's a singlet. While if it inverted, it spins during its excited state for some reason. Yes, we could say that a spin is a spin. Just to explain this, as if it were revolving over itself. If it, for some reason, it inverts its state, then it's called a triplet, and formally it cannot return to the fundamental state. This is the reason why phosphorescent is much longer lasting, because the electron takes much longer. Phosphorescence takes place every time there is an atom, heavy atom in the molecule. I showed you that there were some molecules binding metallic atoms, which are very massive. Iridium, for example, weighs 10, 20, 30 times than all the other atom that surrounds it, and therefore favors phosphorescent. As well as having a different phenomenon, we also have different applications. The one which we see in fireflies is bioluminescence. In the animal world, there's usually one species that uses luminescence. There's an Italian 75 that cataloged them or classified them much more frequently, very deep down in the sea, because that's how they produce light. Usually they use bacteria that have luminescent processes. That is to say, they have chemical reactions, and the product, instead of being at the fundamental state, is at the excited state. Sooner or later, it issues a flash of light, and they use it for various reasons. The fireflies do it to mate, while in the deep, deep sea, in the abyss, for as predators, or simply to show themselves. Right. When you have photovoltaic cells that produce energy from light, and you were comparing also myrtles, and you were talking about, could we imagine that we might use bioluminescence more massively? One of the biggest results in biology is the fluorescent protein from a medusa, and it was extracted from hundreds of thousands. They were fluorescence proteins, and this has opened a new field in biology. Incredible. I don't know whether they will be ever applied to energy or not. Luminescence in nature is something very transient, like fireflies, to have some luminescent moments that the animal uses, while in devices such as these, we want something extremely stable. Silica panels are like stone. Once you've done it, they won't break again. It will go on transforming energy until somebody destroys it. This type of Graetzel cells have a problem of stability, which is not to do with the dye or color, but it's to do with a solution, the fluid part of the cell, which unfortunately dries up and is dispersed. We've created at this point, as well as mine, extremely stable dyes, much more than the ones that fireflies use. Any other questions?
In una lampada a LED a basso consumo, che sostanze ci sono?
I would just like to know, in an LED low consumption lamp, what substances are present? I've asked various electricians, but nobody knows. Yes, it's a question that physicists might answer rather than chemists, but I'll try and answer. What I showed you in the animation, the first application, they were OLEDs. And the O changes everything. O is for organic. That means there's an organic molecule. And that's where you absorb electricity and then it's released. In the LEDs, there's no O. So for you to understand, because they basically are a wafer of a semiconductor, one on top of the other, I hope and think that I have explained how silica panels work. When the sunrise or the light ray creates, there's a dip and then the electrons are injected. We could also go back to it. The LED works like that, but the other way around, exactly like a dynamo, an electric motor. In the case of an LED, there's no photoelectric activity, but it's inversed. So electricity arrives, and where the electron moves or it recombines according to how you see it at that point. What do you mean by a hole? It issues a photon. Well, it creates a photon. Well, the notion of a dip or a hole is a device that we use to explain electricity, because we all imagine electricity as electrons that move. But when we talk about the photoelectric activity, it's easier to imagine holes or dips, which are where the electron can go into. If I have an electron and a hole, then the electron is moved. Here you have a hole, and then so if I imagine here that there's another one, the electrons simply hop from one to the other. I can imagine both the electrons or the holes to move. When they meet, they match. In the case of LEDs, I create a state where a photon is created. The type of semiconductor and electrons and the energy levels will make it possible to create LEDs with colors and so on. This was quite complicated because initially they were only infrared, then yellow, then the green. Whites were very difficult, and then they found two solutions, and the Nobel Prize this year was awarded for the white LEDs. What is organic? That you basically have carbon. Yes. Something which has carbon, hydrogen, oxygen, and nitrogen. I'm more interested in the development of research, and I was wondering whether research started with the idea to find molecules that interacted with light, or having found the molecule, you then discovered that it interacted with light. First of all, he's one of the students of the lab where I'm working, so I would like to acknowledge you and welcome you. As far as I'm concerned, I will tell you, I will confess, I know it's interesting applications, it's interesting, but I'm really no interest in it. I must say, I like chemistry because I like to create molecules. And the satisfaction that you have in the loneliness of the lab, where you solve the puzzle and you understand what's in that powder, is a great satisfaction. That is the reward of research, and is basically the reason why I'm happy when I come to the lab in the morning. But useless things after a bit don't lead to anything, and I realize that. So it's also positive that by obtaining a very high number of different molecules, all with different properties, at a certain point you might ask yourself, for what could these properties be used for? In my case, I was lucky because there are lots of properties that answer a number of requirements. I was talking to Professor Giorgio of the Botanical Gardens. Probably many of the molecules will work for microscopy, so now we will start working together. I didn't know about this, possibly because I forgot what gather the information on what other type of applications there could be. It's pleasure, really. That's why you carry out research. It's like sport. It's because I like it, and I think it's the same thing for my colleagues. Applications and let us say it's applications is never ours, and that's where you get the patents and money. And yes, I agree. I've met lots of scientists who are curious, and it's the pleasure of satisfying your curiosity. At one time, it was called pure or research or fundamental research, but have you patented these things or haven't you? Will you? Is it useless to patent them? Is there some reason why not? Is it technically it's possible to patent it? We're thinking of patenting these. Well, then don't tell us too much how they work. These were maybe one of the luckiest finds, because for me they represented 2.5 months of funding. And the company that had asked us to produce this research now is very interested. A patent, we're talking about 40,000 euros, so no one can pay. So the company or the Department of Chemistry and the company will, we're not here for money, for the money. Well, if not intellectually. Both are fine. I agree. Join the applause, but there's a question here. If part of the income were to come back to the lab and people like you, this wouldn't be go amiss. Something I said on the radio too, the university radio, it's important that companies fund research, but like happens in the US, but companies want a very immediate return. No one would have funded Becquerel when he was 19, he discovered what he did, because there was a very low yield. But if there hadn't been someone, in that case it was the state, the French government, we wouldn't know anything about the photoelectric effect. And the 0.6% yield, we wouldn't have reached the 40% that we now have on the panels that we send on the shuttle. So the state has to fund useless, in inverted commas, research, but maybe in 30, 40, 170 in the case of Becquerel years, it becomes very useful. So I'm in favor of basic or fundamental research. I agree. You mentioned Daguerre, and I think that Daguerre, to an extent, had developed this system to produce photographs. They were called daguerreotypes, and Arago, who was a scientist but was also a politician, to buy it on behalf of France, and a politician who had understood chemistry in exchange for the patent. A question there and a question here.
Buonasera.
Good evening.
Volevo sapere se tutto questo
I'd like to know if all this has to do with the rainbows. Rainbows of light and color, and I'd like to know about rainbows, because I showed the example, the lights, and I'd like to know if there's all this also within a rainbow. There are certainly some connections, because we're talking about colored light, but clearly there are no biomolecules and nothing to do with biomolecules in rainbows. It's a light effect not due to luminescence, but it's also due to diffraction. It's a light on the droplets in the atmosphere, which is an optical effect. That is to say, as if the Vikings tried, if you find the bottom of the end of the rainbow, you'll never find it, because it's an optical effect which consists of very small effects. Nothing chemical, nothing to do with luminescence, but it's a luminous effect, like what you often see in the North Pole, or St. Helm's effect on the ships. It's the interaction of light with matter, but not much chemistry.
Grazie a tutti di essere stati.
Thank you very much.